I am very, very happy to be here. As you can probably tell from the last bit of my name, I'm actually half Greek. I unfortunately do not speak Greek, with the exception of, I can pray, I can eat and drink, and I can get my face slapped. That's about it. But I still feel kind of Greek, and I'm very happy to be here in Athens talking about the science, and I look forward to having collaborations with Greek scientists as well. So, bugs in the brain. What exactly do I mean by that? I mean, they asked me basically to give me like an interesting kind of jazzy titles to the incredibly boring stuff that I do. It's not actually that boring. But so I figured I'd go with bugs in the brain. But that doesn't mean, what kind of bugs am I talking about? I'm not talking about this kind of bug. I don't know if, when I was a kid, there was this Star Trek movie, Wrath of Khan, and they had this disgusting bug that actually they put into a guy's ear, and he would go into his brain and do horrible things. So, the word bug in English means a lot of things. It can mean insects, okay? But, and I don't mean insects. I'm not saying we're going to stick insects into people's brains. But it also means the bugs that make you sick, bacteria, and in this case, actually viruses. So at least, we're not going to be doing this kind of thing. But, that does kind of raise the question, why on earth would anybody want to have a virus put into their brain? I mean, I can see the villagers thinking about actually, coming to my lab with torches and pitchforks, if we start talking about this kind of stuff. But there's actually a pretty good reason. Which is basically, if you're desperate, if you think it's the only way that you can actually fix things that are going on in terms of disorders of the brain. And there are very many disorders of the brain, and some of them are quite horrible, actually. It's about 35% of the whole disease burden in Europe. And it costs around 400 billion euros per year in Europe alone to treat. So it's a big problem. And big problems require big solutions. So there's been a whole lot of effort that has been put towards trying to figure out how the brain works, because it's really hard to fix something if you don't know how it works in the first place. And we still don't really know how it works, even though there's been an enormous amount of funding. Over $10 billion a year, at least, in funding worldwide. And about 1 in 4 of the Nobel Prizes in physiology and medicine actually have been in neuroscience, which is actually more than actually go to people who actually practice medicine. So it's not like we've been totally lazy or we're stupid. But the problem is, we haven't been able to cure anything in the brain, even though there's all this effort and all this money that's being thrown at this. And why is that? Well, basically, it's a pretty simple answer to a very complex question, which is that the human brain is quite simply the most complex object in the known universe. So, what do I mean by complex? Well, it's composed of 100 billion specialized cells that are specialized for communication, called neurons, right? And that's a lot of cells. That's a lot of things. It's basically the same number of stars that exist in our galaxy, the Milky Way. So the numbers quickly become astronomical. And to make it even worse, each of these 100 billion neurons is connected to, on average, around 10,000 other neurons. So the complexity is just insane. I mean, it's truly mind-boggling. There's a biologist who once said, if the brain were simple enough for us to understand it, we wouldn't be simple enough. We'd be too simple to do so, rather. So, here are some neurons, or some rather highly stylized neurons, talking to each other with this kind of little zappy little graphic there. And basically, it shows you that neurons are basically, we are, in fact, basically electric meat, okay? Every bit of our body, every verb, everything we do, everything we think, everything we do is because of a little electrical explosions called action potentials. And this is also the basis for how neurons talk to each other. Again, these are specialized cells that are specialized in communicating with each other. So they release molecules called neurotransmitters. They kind of have these connections between these neurons to basically talk to each other, these things we call synapses. And they basically throw out these neurotransmitters in a tiny little bit of space between them. And then they diffuse over to the next neuron in the synapse. This is the presynaptic neuron. That's the postsynaptic neuron. And they bind to specific receptors to those neurotransmitters, which causes an electrical event to happen, either directly or indirectly. And this electrical event basically causes a response in the neuron. And remember, there's 10,000 or so of these synapses on these neurons. Sound? Did I? Okay, now I can use this sound. So these neurons basically have lots of these, 10,000s of these little synapses on them, each of which, when the cell before it spikes, gives a tiny little electrical potential. And all of these things, some of them make a positive potential, some of them make a negative potential, and they kind of sum up, actually, as they go towards the cell body of the neuron, where the neuron basically decides if it's going to say yes or no. Good. Just keep with this? Okay. So the neurons then say basically yes, in one of these little explosions of action potentials, right? And this action potential then travels down the axon of the neuron, right? And then until it gets to the presynaptic terminals of that neuron. So this guy was postsynaptic to these synapses, and now it is presynaptic to the downstream neurons. So basically, that's how in a very simple way, how your brain and body work. You have these cells that sum up the inputs from lots of different neurons, and then decide whether they want to spike, have an action potential, which then causes the cells downstream, causes the neurotransmitters to release by that cell, and then it goes down to the downstream neurons. So this is basically how all drugs work, whether they are recreational or medicinal or whatever. Okay, what they basically do, most often they basically, they will mimic the activity of the normal neurotransmitter and either do it more or less. Or they can make more neurotransmitters sit around in the synaptic cleft. They just basically amplify or de-amplify, or bring down the amount of communication from one side of the synapse to the other side of the synapse. So, to sum up, basically, the brain is an enormously complex set of interconnected electrical elements. In other words, it is an amazingly complex set of circuitry. So, in this light, you can basically think of nervous system disorders as circuit imbalances. Okay? Well, there's something wrong with the circuit. So here's a very, very simple circuit. It's a circuit that actually has, this microphone has this. It's an audio amplifier. And it has a bunch of different circuit elements, little resistors over here. This is the amplifier itself. And there's a capacitor over here. There's a battery. And basically, if you put, the way we figure out how circuits work, how engineers figure out how they work, is they take this little element, this single element, and they put a potentiometer, or a pot, on it, right, that allows them to turn it up and turn it down. And as this pot goes up and down, you'll actually hear the sound goes up and down itself. Now, the problem with this is you absolutely have to manipulate a single circuit element at a time. Okay? If you did a whole bunch of them, it wouldn't work at all. So if the brain's the most complex circuit alone, then what you have to do is kind of find the proverbial needle in a haystack. Okay? You have to look around and see what exact part of the circuit is going wrong. And your manipulation has to be specific to that circuit. So right here, you can see this tiny little red X. That's the little circuit element that's out of balance in this particular circuit. And this is the one we want to manipulate. And this is true for both the science of neuroscience, and it's also true for the eventual, it's the way we have to treat neurological disorders. You can't just take out a huge chunk of the circuit, right? Or manipulate a huge chunk of the circuit, ignoring all of the interesting little circuit elements that are there in that piece of the circuit, right? That's basically what brain surgery is like. You're not differentiating between all the neurons that make up the circuits and the part of the brain that you're actually going in and scooping out, or destroying with radio waves or whatever, however you want to do it. You are just taking the whole circuitry and removing this big gross amount of it. Sometimes it works very well, certainly better than nothing. Or you could give a drug, which again, as I said, the way drugs work is they basically activate certain receptors at the postsynaptic side, right? Or they deactivate them if they're either agonists or antagonists of that receptor. But the problem with this is, this receptor is not just in one circuit, it's in many, many different circuits, some of which are unrelated to the actual neurological or neuropsychiatric issue that's going on. A lot of little X's all throughout the circuit. Doesn't work. So what can we do? Does it work? So y'all can hear me through this? Good. Great. I walk around a lot. Thanks. So, well, these are actually pictures from one of the very first Nobel Prizes. This was from Ramon Santiago y Cajal, a Spanish neuroanatomist. And he saw, it was actually kind of funny, you can, if you're interested in the history of science, he shared the Nobel Prize with Camillo Golgi, who was the father of cell biology, did amazing things, made all these wonderful dyes. And he basically made this silver stain that we still call a Golgi stain. And he looked at all kinds of things with it, including the brain. And that's all that Cajal did. He looked at the brain using Golgi's silver stain technique, but the difference was he realized that it was circuitry. He realized that basically, and he actually even understood the information flow, the direction of information flow through all these different neurons. And there's so many different kinds of neurons. We actually don't even know how many different kinds of neurons there are in the brain. So in a hunk of brain, right, you have all this different circuitry. We need some way to actually address both our scientific inquiry and our manipulations, and indeed, eventually, our therapeutic manipulations that have this level of specificity. Affects these cells, but not those cells, for instance. Getting beyond the bolus and beyond the making of systemic injections of systemically active drugs. So I have to give you a quick primer on molecular biology, because the only way we can actually address treatments and studies to the circuitry of the brain at the level it actually operates is by kind of co-opting the genetic machinery that gives it this complexity. And we can do that through molecular biology techniques. And basically, you know, it's not that complicated, molecular biology. If you know, DNA is DNA is DNA. It's like Mother Nature's text, right? Except it's spelled G-A-T-C-G-G-G-G-T-G-G, right? So just like text that you write, you can sometimes cut and paste it, you know? Take a little bit of this and stick it over here, and take a little of this and stick it over there, and you basically, sometimes it works and sometimes it doesn't, just like when you're writing something. So that allows us to take things from, do all kinds of stuff you read about in the newspaper about genetically modified organisms. You can take a gene from a fish and put it into a strawberry, and you make strawberries that don't freeze. You can do all kinds of crazy things with this. But what you can also do is manipulate the genomes of viruses to make them tools, actually therapeutic tools, something called gene therapy. So what we've been doing in the lab is, since we're a physiology lab, we're interested in this kind of circuit breaking thing, and especially our special interest is actually in memory. But we realized we needed to make molecular tools that were very, very specific for specific cell types, so we could go in there and do a little circuit breaking. Turn them up, turn them down, see what happens to the rest of the circuit. So I have to tell you a little bit more of molecular stuff, which is basically what is gene expression. Basically, DNA gets copied into messenger RNA, and that messenger RNA gets made into proteins. Okay, a process called transcription. So this transcription is extraordinarily regulated. And it turns out that this regulation of transcription is it's relatively simple in bacteria, where it was first studied. But in eukaryotes, basically things with nuclei, what happens is there is still, like in bacteria, this core promoter, where the RNA polymerase, the enzyme that copies the DNA into RNA, binds. But way far away, there are these other bits of DNA that other proteins bind to that basically tell the promoters when and where to express the gene. Okay? Now, there's only 42,000 or so promoters. There's only 24,000 genes, right? And there's way more. Nobody really knows, okay? But estimates of how many different kinds of neuronal cell types that actually exist are often way over the number of genes and the number of promoters. And indeed, most promoters express throughout the brain, or in many different cell types in the brain. So how can you get more specific than gene expression itself? Well, it turns out these enhancers and repressors, these other things, these other parts of the genome that tell the promoters actually what to do, where and when to make the protein that they make, that gene makes, to express that gene, basically nobody really knows, but the estimates go up to about 2.5 million different constrained elements. So there's plenty of molecular complexity to use to be able to, make some interesting therapeutic toys. So, long story short, we started working on this and trying to make artificial genes, in a sense. If a gene's basically a control region and a payload, right? We take some of the control regions. Usually there's hundreds of these different control regions that talk to the promoter, and then we take a promoter from a virus and we stick them together, just one of them. And what we find we get, and we do this over and over again, this is actually not published yet, but it will be soon, is a gene that expresses throughout the brain. These are mouse brains, cut sagittally, cut like sideways like this. You actually see that you can, by using just one of these control regions of a particular gene and making a synthetic promoter with it, you actually get a gene that expresses all over the place to express in very specific cell types. Again and again, lots of different cell types in a targeted manner. You actually can target which part of the brain you want to do this on. So, why should you care? Well, again, basically, brain disorders are circuit imbalances. And drugs, maybe, they might do the right thing in the right circuit, bring it up some. But they often do the wrong thing in the wrong circuit. That's why all these drugs have such side effects. If you ever see a psychiatric patient, they often have lots to say about the side effects that are happening. And what's happening in these side effects, again, is that the drug is binding to the receptors that are out of balance, but it's also binding to the receptors elsewhere in the brain. So what you want to do, if possible, ideally, is first understand all of the various disorders of the brain in terms of the imbalances in circuits that they are. And we know this for only very few disorders. And then try to design a virus, a bug, right, that is tailored to going directly to that particular neuronal cell type, and then have a therapeutic transgene, something that you express that allows you to fix the problem, okay, and only in those cells. So that would then treat the disease without causing the side effects. So I'll go through a couple examples now, just to show how this can be done. It's not being done yet, but we are building the tools in my lab now. We're getting there, actually. So maybe some of you have heard of Parkinson's disease. It's a really horrible neurodegenerative disease. It has happened to Muhammad Ali, Michael J. Fox, and other relatively famous people. And you can see they start to shake. Actually, they have what's called a resting tremor. They just which is basically you can hear the clock of the motor system in these folks, because it's an imbalance in motor circuitry and the circuits that are involved in initiating motor actions. So they end up having this resting tremor. They're very stiff, right? They can't initiate movements, but they're always kind of tremoring. And a mask-like face, they say, right? It's they always have the same expression on their face because they're taut, basically. And differences in the voice. You can also get it, actually. These folks, MPTP patients, they actually got Parkinson's because they were trying to cook up amphetamines, and they didn't cook so well, so they actually poisoned a particular part of their brain. Very specifically, the dopaminergic neurons of the substantia nigra pars compacta. So substantia nigra is, I mean, it's a very fancy sounding word. It just means black stuff in Latin. And what you can see, if you were to transect a Parkinson's patient, obviously you do this posthumously, you'll see that in a normal person, you can see right at the brain stem there, it's actually the midbrain, the substantia nigra, this black stuff looks very black. And in a Parkinson's patient, you don't see a lot of that black stuff. So this isn't just a region, okay? It's actually a specific set of neurons. And they're being labeled here by the biosynthetic enzyme for dopamine, which is a particular form of neurotransmitter, okay? So you can see that there's other neurons all around these guys, right? This is from a rat. But these are the ones that are dopaminergic. These are the ones that are degenerating, okay? So what this means at a synaptic level, right? Again, oversimplified, but you know, it's not that oversimplified. Normally, the neurons downstream are getting a certain amount of dopamine, right? But in a Parkinson's-affected neuron, you're getting much less release of dopamine. So this particular circuit, the substantia nigra pars compacta to the rest of the basal ganglia, leads to this motor disorder. So how do you treat it? Well, you can, there's actually a lot of strategies that people can do to behaviorally kind of make it seem better, at least. But they get L-DOPA, which is a precursor of dopamine. So they get a lot more dopamine. You just basically add more of the neurotransmitter. But this has some serious side effects because, again, these are not the only dopaminergic neurons in the brain. And there's lots of receptors for dopamine throughout the brain, so you're flooding the whole brain, including the part that's not screwed up, with dopamine. So there's also, you get this receptor deregulation too, so you can only do that for a while, even with the side effects. So what people do if they're really late stage Parkinson's, pretty much the only thing you can do is deep brain stimulation, which means you open the person's skull and you implant a stimulating electrode. Remember, all this stuff is electrical. So if you put electric current in there, you activate neurons, right? So you put a stimulating electrode into the brain, bring it down to either the substantia nigra or areas that are talking to the substantia nigra, or the pathways, and you do high-frequency stimulation there. And you should look at it on YouTube. It's amazing. When it works, it's amazing. It's a cure. But the problem is, what it's doing is it's creating more activity of these neurons which are degenerating. Okay, so it's fixing the imbalance in the circuit. However, the problem with these patients is that, well, electrical stimulation is going to fry the brain, literally. It's like frying an egg. You have to keep adjusting the electrodes on these patients because you actually fry the tissue that they're stimulating around, and so it's only a temporary measure. And beyond that, you have a wire going from the outside through the skull inside, which wicks bacteria underneath the skull, and they have these horrible infections which often actually kill them, unfortunately. So how much better it would be, what we're trying to do with deep brain stimulation is stimulate these particular neurons, substantia nigra, pars compacta, and you put an electrode in, you're stimulating everything. Remember how this looks. We want to just stimulate these guys, right? How much better to use some bugs? If we can tailor these bugs to only express their payload, a therapeutic transgene, in the neurons of interest, in this case substantia nigra, pars compacta, then we can add the therapeutic transgene. So you start with a therapeutic transgene. I'll show you what that is in a second. Wonderful thing. They're called DREADs. And you basically take that transgene, you put it into a recombinant adeno-associated virus, AAV, right? It's a relatively nice virus, as viruses go. It can stay inside the cells for years, actually, without actually hurting them. And you can inject it into the blood, okay? And some colleagues at Caltech actually have just recently figured out a way to put a protein on these viruses so that they cross the blood-brain barrier, which is something that everything that's going to get to the brain has to do. So you don't have to even inject into the brain anymore. You just inject into the blood, they infect a bunch of neurons in your head, and they end up infecting and only expressing their payload in the neurons that have been screwed up. And what their payload is, it's a beautiful thing that Brian Roth at University of North Carolina, Chapel Hill, has come up with, are DREADs. He has a flair for marketing, too. DREADs are designer receptors exclusively activated by designer drugs. And we don't mean club drugs by designer drugs. We mean actually, it's a drug, basically. Remember how drugs work? They basically bind to and activate or deactivate receptors, right? So this is a neurotransmitter receptor that's mutated to no longer respond to its natural neurotransmitter, okay, which is acetylcholine in this case. But instead, it responds to this drug which has no binding affinity for anything else in the brain. So you can basically turn up the neurons or turn down the neurons, depending on which DREAD you want, just by giving a systemic, just by taking a pill, basically. So for Parkinson's patients, if we had a viral payload with a virus with the tropism for substantia nigra pars compacta neurons, instead of doing deep brain stimulation, brain surgery, you could just inject this into someone's arm, goes to the right neurons, and then the patient takes a pill to basically fix their Parkinson's disease. Another one that I think at least, and we're working on this now, is a disease, a horrible disease. I've seen it happen to my mother-in-law, actually. Is a neurodegenerative disease called Alzheimer's disease, which is a dementia. There's been an enormous amount of work done on this, and basically it's a progressive disease that has a long asymptomatic stage. Okay? People have Alzheimer's for a long time before you can actually diagnose them as having Alzheimer's. They're in the early stages are undetectable is the problem. So this is a problem because by the time you get diagnosis of Alzheimer's, it's almost too late to do a lot of the interventions. It's like a volcano. The asymptomatic stage, basically, you can't diagnose. But there's something going on in the asymptomatic stage. And then, basically, it starts in certain neurons, it turns out, and then jumps across. Kind of like viruses, you know. It actually starts somewhere, and then as it goes further and further into the brain, the patient gets progressively worse and worse. The reason why patients start off with cognitive and memory deficits, okay, is because, remember I told you my lab does memory research. The neurons where it starts, what they call the pre-alpha neurons, for the clinicians, are in this area called the entorhinal cortex, which is the input to the hippocampus, which is basically where memories are made. So but they don't die of lack of memory, actually. They can live for years with their dementia. But eventually, since it spreads transsynaptically, it knocks out parts of the brain that eventually will kill them. So what we need to do, there have actually been very successful clinical trials in the sense that they've removed the plaques that are associated with Alzheimer's, but they couldn't remove their symptoms because they got in too late. So these very neurons, again I said, are in the brain region that we study. In fact, they are these neurons. They are entorhinal layer 2 reelin positive calbindin negative projection neurons, which is, if you're a neuroanatomy geek like myself, you know, are these guys here. So what we've done, kind of a proof of principle thing, is we've taken the bits of genetic material that we had in these viruses, in these mice that we used to make these mice that express just in these neurons, these pre-alpha neurons. And we've put it into an AAV and injected it systemically. And we find that we only get expression. The green guys are the neurons that are expressing the virus. We can target directly these neurons that are the first neurons to go, basically, in Alzheimer's disease, with the idea that if we can then target these neurons with something that slows it down, there's a large number of possible candidates. Alpha secretase is one of the ones we're doing. We're still doing this in mouse models, right? You should be able to actually start to slow the disease progression down because it's transsynaptic. So you kind of catch it before there are symptoms. So that's basically what we're up to. I'm happy to take questions if this makes any sense, and please don't come after me with torches and pitchforks because thank you.